All right, so this is the recording for the last Biology 120 lab of the semester, Lab 14. And in this lab, we're looking at animal phylogeny. So very similar to what we did earlier in the semester with the plant phylogeny lab. We're going to do the same thing, but looking at different animal groups, specifically Bilateria. And we're going to use cladistics like we did before to figure out which is the most parsimonious tree, which is the best tree representing relationship between these animal groups. So similar to what we did before, you're going to choose um, 6 or more traits that are shared by some but not all bilaterian animals. So like I mentioned in the previous lab explanation, if you choose a trait that's unique to one group of animals only, then that actually won't factor into the cladistical analysis because it's always going to just be on that one lineage. If you use a trait that's found in all of them, that also won't matter because in all of your trees you come up with, all of the groups that you're considering will all share that trait. So the best traits to use for this are traits that are shared by some but not all members, so more than one. And I gave you some traits to consider. You don't have to use any of these. You could think of your own, but you're welcome to use some of these. So body cavity type, which would be, are they acoelomate? Do they have a pseudocoelom or a coelom? And I mentioned before that this isn't an absolute trait, but it is a trait that is relatively conserved, so you could use that as one of your traits. The presence of segmentation, type of symmetry, radial, bilateral symmetry, gut type, do they have a sac-like one-way gut, or do they have a tube-like gut that has, a separate mouth and anus, complete digestive tract? Respiratory system type, do they have a respiratory system within that group? Do they have gills? Do they have lungs? What type of habitat would they be found in? You could say, what's the predominant habitat, or do they have any members? For some of these animal groups, none of the species within that group live on land, like echinoderms, for example, are all just in marine ecosystems. So you could use habitat as one of your traits. Skeleton type, do they have a hydrostatic skeleton, an internal kind of bony skeleton, or an external exoskeleton? That's another trait. Do they grow by molting, by kind of shedding their outer layer? What type of circulatory system do they have? Do they have one? Is it open or closed? Reproductive pattern, you could say, are most species of this group hermaphrodites, or are they mostly separate sexes? I didn't do that trait analysis here, but that could be something that's interesting if you look that up. So again, just pick 6 traits. You could use some of mine. You could come up with your own. So the first step, again, is to choose 6 or more traits that could be used to separate these different animal groups. And this assignment is actually simpler than when we did the plant phylogeny lab, because in this case, you're only comparing 2 trees that are given to you. So these are figures 1 and 2 from the lab manual, but I've reproduced them here. So you can look in the lab manual, or if you just want to watch this video to figure out what the trees look like. I can kind of tell you what these trees represent. So this tree on the left here, we have quinoflagellates, which are a sister group to animals, and then we have porifera here, the sponges. This is not showing all of the groups. So for example, placozoans are not here. Cnidarians would be here, and then all of these guys would be the bilaterian animals. So in this tree, we have platyhelminthes kind of off by themselves, and then roundworms, and then we have arthropoda and annelida, the segmented worms and arthropods together, and then mollusca. And then, similar to what we had before, echinoderms and chordates are together. So this tree looks very different than the tree I presented to you during the class, and that's because this tree is wrong. It's an old tree. If you looked at a textbook from I don't know, 30, 40 years ago, it might look a lot like this. So this is what people used to think, but it's wrong. If you include a lot of traits, if you include DNA analysis, this is not a good tree. On the right here, this is the tree that I presented to you, which is, by all indication, by all the data we've collected, many different phylogenetic analyses, this is a really good solid tree, much better than the one on the left. And this is the tree I presented to you. So we have on the right here the deuterostomes, echinoderms and chordates, just like we had before. On the left here would be the protostomes as a monophyletic group, broken up into the ecdysozoans, which would be the roundworms, nematodes, and arthropods. There's also other groups here that are not shown. And then the lophotrochozoans, which would be on the left here, the flatworms, segmented worms, and then the mollusks. So that's pretty much exactly the tree I showed to you. And what you're going to do is basically use the traits that you select to compare these two trees to see, does your cladistic analysis match closer to the tree as we know it today, or does it match more closely with kind of this old tree, which has been disproven? And so these are the groups you're considering that you want to find the relationship of: flatworms, roundworms, arthropods, segmented worms, mollusks, echinoderms, and chordates. And the way you're going to compare them is just comparing these two trees. For both of these trees, you'll see that the position of these outgroups is the same. So quantiflagellates is on the outside, and then sponges, and then cnidarians. So those positions for those three groups are the same in both trees, so you kind of don't have to worry about them that much. And you can kind of treat cnidarians, because cnidarians would be the most closely related to the bilaterians as presented here. So cnidarians can be your main outgroup. You can use them, and that's what I showed you in the table as part of this video, is what are the cnidarian traits. If you wanted to, you could also include poriferans and quantiflagellates, but it could get a little bit more complicated that way. But basically in both of these trees, it's agreed upon that cnidarians, sponges, and quantiflagellates are outside of the groups that you're considering. So they are going to be outgroups in both trees. Okay, so the second step is basically to take the traits that you identified, and then you fill out a trait table. So for all of these different animal groups, which is 7 different animal groups, along with, I included cnidarians, which was the major outgroup that I think you could use, because they're the most closely related to these 7 groups. There they are in the table, and then I just picked some traits, and I filled it out for you. So if you want to use some of these, don't use all of them, because I want you to do some work for yourself, but you can use some of these. So I basically show you symmetry, everything's bilateral except echinoderms are radial and cnidarians are radial symmetry. Segmentation, kind of spotty. Flatworms, tapeworms, again, are segmented, but most of them are not. Segmented worms are segmented, obviously. Arthropods segmented, chordates segmented. So some of the groups have segmentation. About half have it, about half don't have it, that's a good trait to use. Respiratory system, some of them have no respiratory system, dedicated respiratory system. Cnidarians, roundworms, flatworms, no respiratory system. Some groups just have gills, like echinoderms just have gills or something that operates like gills. Segmented worms, some of them have gills. Most of the groups don't have anything, any respiratory system. And arthropods and mollusks and chordates, you have the existence of gills and lungs within those groups. So you might want to think about how you're going to group these. You could group it in terms of, do they have respiratory systems at all within the groups? That could be a way of doing it. Or you could also look at, do any of the species within this group have lungs? You could do that as a characteristic as well. I also did this because I thought it was kind of fun to look at maximum size, and I think this is accurate. There are some species where I'm not sure exactly how much they weigh, but I think this is accurate. So basically, the biggest member of each group, would it weigh more than or less than 100 pounds? Which is kind of fun. You could do a trait like this. It doesn't have to be something you learned in lecture. You could just do something fun. So based on this, I think that the only groups here that would have really large members like this would be the mollusks, having giant squids, colossal squids, even giant clams, I think, are heavier than that. Chordates, obviously, there's very large vertebrates, and also cnidarians. There's some really heavy jellyfish, like the lion's mane jellyfish is more than 100 pounds. But the other groups, I'm pretty sure, arthropods used to have members over 100 pounds, but I don't think anymore. Echinoderms, no. Segmented worms, I don't think anyone else has really large species. I also did flying. There are, arthropods, some of them fly, chordates, some of them fly. So that's another trait. A couple groups share this. So all of these traits I've listed so far would be kind of good. The kind of the worst trait I have here is symmetry, because only one of the members of the group, echinoderms, has radial symmetry, but the outgroup also has radial symmetry, so you could still use that one. Skeleton, a lot of these have hydro skeletons. So basically that, they use some type of fluid pressure to have muscles exert against, hydrostatic skeletons. And then there's exoskeletons in arthropods, and then endoskeletons in chordates and echinoderms. Limbs, I'm not sure if this is completely inclusive. As far as I know, I don't think flatworms or roundworms, there's any examples that have limbs, and then everybody else at least has some limbs. And, it kind of depends on how you define limbs. But anyway, that's a table. You're welcome to use some of these if you want to, but it's kind of fun to create your own traits and try and fill it out. So that's the next step. Make a table similar to this. Okay, so the next step is, this is kind of similar to what we did for the plant phylogeny lab, is to figure out ancestral traits. And the reason you do this is, in a sense, it kind of doesn't matter what you think the ancestral trait is, because in a lot of cases, if you said the ancestral trait was one way or another way, you could do the cladistic analysis any way, but it allows you to kind of keep track of trait changes, because you're imagining, okay, this is what the ancestor looked like. Any deviation from this would be considered a trait change. So that's why it's useful to make this table. So basically, look at your table of traits, including what would be present, among the outgroup, or if you included a few outgroups, that's fine too. And then figure out, okay, what was this ancestor like? So, this ancestor, one of these ancestors that kind of gave rise to the Bilaterians, what were their characteristics most likely like? And it's okay if you're wrong, you're just trying to come up with something, with a reasonable deduction of what this ancestor was like. So what was the ancestral characteristics before these splits between the different Bilaterian groups? So a good kind of rule of thumb for this would be, if some of your animal groups you're looking at share a trait that's common with the outgroup, then that's a good candidate for the ancestral trait. So for example, cnidarians, sponges, they don't have segmentation. And there are groups of bilaterians that don't have segmentation. So that increases the likelihood that, well, maybe the ancestral trait, before the split among bilaterians, was not having segmentation. Same with no respiratory system. Cnidarians, sponges don't have respiratory systems. Several groups of bilaterians also don't have respiratory systems. So not having a respiratory system would be a reasonable ancestral trait. Having a hydrostatic skeleton would be a reasonable ancestral trait. So basically make a table, another table, for those 6 or more traits you chose. What do you deduce to be the ancestral condition for those traits? And then any changes from those ancestral conditions in your tree leading up to your 7 animal groups will count as an evolutionary trait change. Okay, so now that you have your ancestral trait table, you're in a position where you can basically compare your ancestral trait table to the groups of animals as they exist today and map where changes occur. So for example, if you said that the ancestral trait was not being segmented, right, then any incidence of segmentation within those 7 groups would have to be marked in your tree. Now, what groups are segmented? Well, chordates are segmented, segmented worms are segmented, and arthropods are segmented. So you'd have to mark basically evolutionary transitions to allow for those groups to be segmented today. And follow parsimony. So if a change can involve only once rather than twice in your tree, then map it as a single change. So you can look at the videos for this, but as an example, again, with segmentation, if the segmented worms are sisters to the arthropods, right? If they're on two sides of a split, they're both segmented. So the existence of segmentation in those two groups, you can mark that as just one change towards segmentation in one of their common ancestors along the lineage before they split. You just basically mark segmentation once, and then that would allow arthropods and segmented worms to both maintain segmentation. If arthropods and segmented worms are distant in your phylogeny, then you'd have to mark those as two separate evolutionary occurrences. And I know I've talked about this before. If you're still having difficulty with cladistics, with mapping trait changes, with counting them, see the previous videos I posted on YouTube. I have my lecture on phylogenetics. I have the video that I did for the lab, for the plant phylogeny lab, and also I did the group activity review where I talked about how would you actually do the group activity assignment with those little creatures that I created. So if you're still having trouble, I would recommend just watch those videos again, and hopefully it'll start to make sense to you. But if you're still having trouble after rewatching those videos, let me know and I can try and help you. But do the best you can with the materials that I've posted already. After you map all of the trait changes onto your two trees, because again, you'll have two trees this time, then you're going to count up those trait changes for each tree. Okay. So based on, by cladistics, the tree that has the least number of changes, the least number of evolutionary changes, is the better tree. That's the most parsimonious tree. It's the simplest tree. So that's what you would choose as the most likely tree. So in the online assignment, which I've created to go along with this lab, you're going to upload a document, some type of document file, or it could be a picture if you take a picture of, a written assignment. Any of that's fine. It's up to you. Just somehow represent this. You should have 4 things. So you should have your trait table filled out. So for those 7 animal groups we're looking at and your 6 or more traits that you choose, fill out that table like I showed you. And then have the ancestral trait table. So for all of your traits, give an indication of what do you think the ancestral condition of those traits would be. And then you should have a drawing of your 2 trees, and you could also just print it out from the lab manual. There's pictures there, but whatever you want to do. Have the 2 trees, the 2 different trees, and map the trait changes onto those trees and count them up. So, it's nice if you just have a number beside the tree somewhere that says, this has this many total changes. And then give some indication, circle it, or just write out which tree is the best one according to your cladistic analysis. And if you end up supporting the tree that's actually not true, that kind of crappier tree I showed you, that's perfectly fine. You're not going to be penalized. Depending upon what traits you choose, some traits will support one tree and other traits will support the other tree. So there's nothing wrong with kind of supporting the wrong tree. Because if you did this with 1,000 traits, including many different genes, you would end up going with the more accurate tree. But for this, you're just choosing a few traits. So it's perfectly fine to end up getting kind of like the wrong answer here. So that's my explanation. Hopefully you've been following along with this. This is the last lab, and I'll send out some more emails soon, kind of describing more information about the final exams as well as the research paper. Okay, so good luck on everything, and I'll talk to you again soon.